Good morning. Welcome to Sakshi Eapcet Botany question paper answer key discussion. So let us discuss one after another every question and find out the appropriate answer. Question number one, identify the mismatch. A. Cholesterol is a sterol, lecithin is a phospholipid, deoxyadenylic acid, nucleotide of RNA, NAD, a cofactor. So out of all these things, deoxyadenylic acid is a nucleotide of DNA but not RNA. So C is the mismatch. Then the next question. Second question. Which of the following is a toxin? Lectin, resin, anthocyanin, vinblast. Out of these four, all are secondary metabolites, but resin is the toxin. Aberrin and resin, these two are the toxins. So answer is B. Now the next question, that is, flowers are colorful and emit smell when they are wind pollinated, insect pollinated, bird pollinated. More than one option is correct. So the best option is insect pollinate. Insect pollinate. So B is the correct answer. Going for the fourth question, study the following statement. Archaea and eukarya are more closely related to each other than bacteria. RH Whittaker placed unicellular eukaryotic organisms in Monera. Carl Woos replaced kingdom Monera with two kingdoms, bacteria and archaea. 24S rRNA is used as identification of microbes. So we have to find out what are the incorrect statements. Archaea and eukarya are more closely related to each other than bacteria. Yeah, that is correct. Whittaker placed unicellular eukaryotes in kingdom Monera. Not Monera, it is Protista. So this is wrong. Carlus replaced kingdom Monera with two kingdoms, bacteria and archaea. No, this Carlus two kingdoms, bacteria and archaea is correct. Now, what is the left out one is 24S rRNA. It's not 24S rRNA. It is actually 16S rRNA. So, incorrect are B and D. Fourth option. Next. We can go to the next question. So, now coming to the next question, that is question number 5. One of the following cannot carry out nitrogen fixation. Glomus, Anabina, Rhizobium, Frankia. Glomus is actually a fungus that is involved in phosphorus absorption. Phosphorus absorption, but it cannot carry out nitrogen fixation. So the answer would be A, glomus. So the next question now. The extraction of DNA fragments from agarose gel matrix is called elution. The extraction of DNA fragments from agarose gel matrix is called elution, which is the second option. B is the correct answer. We can go to the next question. Vascular cambium in dicot root is formed by Vascular cambium in dicot root is formed by partly from cells of connective tissue and partly from cells of pericycle by dedifferentiation, partly from procambium and partly by differentiation, partly from pericycle and partly from endodermis by dedifferentiation, redifferentiation of pericycle. So here the answer is partly from cells of conjunctive tissue. This is JU. Partly from cells of conjunctive tissue and partly from cells of pericycle by dedifferentiation. So if you take the dicot root, you will be having 4 xylem strands and you will be having 4 phloems. So now you will observe that from here the cambium starts and it will reach the pericycle. So once again it will be like. So the cambium comes from 2 areas, that is cells of conjunctive tissue and cells of pericycle by dedifferentiation. The seventh one, first option is correct. Now going for the eighth question. Which of the following is not a component of bark? So what is actually bark? Bark is periderm plus bast. So wood will not be a component of bark. Wood is inner side of the vascular cambium. That is, wood will not be a component. The third option, C, is correct. Now coming to the ninth question. In ABO blood group, the blood group that is an example for codominance. The blood group that is an example for codominance. Codominance will be seen with AB blood group because both alleles exist, then both express themselves. So C would be the answer. C would be the answer. Now, which of the following facilitates opening of stomata? So if you want to open stomata, the guard cells have to be turgid. Turgid guard cells in the sense they should be having turgid guard cells in the sense they should be having more amount of water. When they get more amount of water, actually the water potential value in guard cells will go on increasing. The water potential value in guard cells will go on increasing. So now here we can keep which option in the sense B as an answer. B as an answer. Now coming to the next question, that is 11. Cells of region of root show protoplasmic modification, attain maximum size in terms of cell wall thickening. See, the above characters of protoplasmic modification attain maximum size with growth are seen for the region of maturation, because different tissues and cell types, they also can be seen in the region of maturation only. So C is the answer. Now the next question, question number 12. We have to see the correct sequence of events in meiosis 1. Bivalent formation, segregation of homologous chromosomes, crossing over, formation of two haploid nuclei. A option is not appropriate because crossing over occurs before segregation of homologous chromosomes. Bivalent formation, crossing over, segregation of homologous chromosomes, two haploid nuclei. This looks to be correct, but let us check the other two options. Coming to C, segregation of homologous chromosomes, crossing over, bivalent formation, two haploid nuclei. So bivalents are formed, then crossing over occurs, then segregation. So this cannot be correct. C. Crossing over in the starting also cannot be correct. So A, B, C, D are not correct. Therefore, B would be the correct. Going to the next question. Number of recognition sites present on PBR 322 for different restriction endonucleases. So what are the different restriction endonucleases having recognition sites on PBR 322, PVU1, PVU2, PST1, BAMH1, SAL1, okay, BAMH1, SAL1, PVU1, PST1, PVU2, ECO R1, CLAV1, and HIND3. So totally how many we are getting in the sense? 8. 13 question answer is first up. Now let us go for the next. Now going to the question number 14. This is what is question number 14. Which of the following is formed in more quantity in intracellular secondary growth from vascular cambium? So if you take the vascular cambium, the activity of vascular cambium is more towards inside, less towards outside. So more amount of food is formed than the outer phloem, the secondary phloem. So the answer here would be secondary xylem, which is otherwise called as wood. That is second option. Sequential filtration. Sequential filtration is a process in sewage treatment used to remove. Sequential filtration is a process in sewage treatment used to remove. So this occurs in the primary treatment. It is used to remove floating debris. It is used to remove floating debris. That is one point. Now the answer is B. Going for the 16th question. 16. Stability of DNA to heat. Stability of DNA to heat as a property was proved by. Stability of DNA to heat as a property was proved by Griffith experiment, Hershey and Chase experiment, Ivory McLeod McCarthy experiment, Messelson and Stahl experiment. In this, the answer is Griffith experiment. How? In the sense, when heat killed a strain, when heat killed a strain is just mixed with R strain, then what happened is the R strain is also converted into S. Even upon heating, the pathogenic nature of the organism externally is lost, but internally the pathogenicity is still retained in the heat killed S strain. So whichever is the substance, it may be DNA or any substance which is responsible for moving from or converting R strain into S strain that was stable to heat. It has not lost its virulent nature. So this is proved by Griffith expert. So answer is A. Epigeal seed germination is seen. Cotyledons are brought above the surface of the soil. So option A is. We can go to the next question. Now, number of reproductive and vegetative characters of tree plant of pea plant respectively studied by Mendel. So Mendel has studied 7 pairs of contrasting characters, in which only one is the vegetative character, that is height of the plant. Remaining all are reproductive characters. So first, what is asked? Reproductive. Next, what is asked in the sense vegetative? So the answer will be nothing but option A. Now, question number 9. Dioecious sporophytes are dioecious sporophytes. The answer comes to be cycas. Cycas is a plant where you will observe dioecious nature. It is not monoecious, it is dioecious. So answer is first option. So now we have to study the given things and choose the correct combination. This is a set of 3 statements. So Km value, Km means Michaelis-Menten constant, indicates the affinity of the enzyme to the substrate. It's the inverse measure of affinity. Yeah, of course. If Km value is more, affinity between enzyme and substrate will be less. If Km value is less, affinity between enzyme and substrate will be more. So Km value is inversely proportional to affinity between enzyme and substrate. So that's why it is written as inverse measure of enzyme affinity. Hydrolases, PO, linking two compounds. No, they don't link, they break. Transferases, transfer of a group. Inhibitor of a reaction. So it's not inhibitor of a reaction. Emil Fischer lock and key hypothesis, formation of enzyme substrate complex. So the correct options are actually first and fourth. So the answer would be 1. You can go to the next. Now question number 21. Which of the following statements is incorrect with respect to factors affecting photosynthesis? Light is a limiting factor for photosynthesis in case of most plants. Second statement, CO2 saturation point for C3 plants is beyond 450 microliters. Increased CO2 concentration for C3 plant like tomato and bell pepper increase rate of photosynthesis. Water affects indirectly the process of photosynthesis. Out of these 4 statements, A, B, C, D, which is the incorrect one? How? In the sense, leaves of the plants show saturation for 10% of full sunlight. Clear that for majority of the plants, light is not a limiting factor, except for those plants which grow in dense forests and also in shade place and also in shade place. For these two, they grow in dense forests and also in shade places. There it can be a limiting factor. For most of the plants, it is not at all a limiting factor. So option A is. Can we go to the next? Now going for the next question. That is, now let us. One of the following amino acid is an exception for the degenerate nature of the genetic. Degenerate nature in the sense, an amino acid should be coded by more than one. This, which is not having that degeneracy property in the sense, which one is D. So the answer is D. Now the next question. One of the following is sometimes present in a bacterial cell: capsule, cell wall, cell membrane, genophore. Out of these four, cell wall, cell membrane, genophore are compulsorily seen in all bacterial cells. Capsule is a layer on the outer side of the cell wall. It may be loose sheath-like, then it is called slime layer. If it is thick and tough, it is more or less called capsule, which may be present or absent. That is the one. Next. So we have to choose the correct match here. When we choose the correct match, let's see what would be the point. Mango, Mangifera indica, true. A with 4 and 3. Have a 2 read. Citrus sinensis, Hesperidium. So B with 3 and 1 is in Dolichos lab legum. C with 1 and 4. C with 1 and 4. Custard apple, Ananas squamosa, aggregate. D with 2 and 2 would be the answer. C would be the number 20. Physiological problems in a plant are because in other particular options, Sphagnum, it shows haplodiploid. Pyrogyra, it shows haplontic. Chlamydomonas, it shows haplon. So the answer is puke. Now, if a polypeptide chain has 99 amino acids, then the number of nucleotides in mRNA, then the number of nucleotides in mRNA will. So mRNA, in the sense, the formula is 3n plus, where n corresponds to the number of amino acids. So 3 into 99 plus 3. That comes to be 300. So answer is D. Answer is D. Now going for the next question. Just a second. So going for the assertion reason question. 28. Pollen grains are well preserved as fossils. The exine of pollen grains is made up of sporopollenin, which can withstand high temperatures, strong acids, alkali, and no enzyme can degrade it. So here A and R are true, and R is the correct explanation for A is the appropriate answer. The first one is the correct one. We can go to the next. Now coming to the 29th question, which is the match the following one. If you just see the match the following, blue dye comes from indigofera, yellow dye comes from butia. So A with 3, B with 5. A with 3, B with 5. Yeah. Medicine is supposed to be belladonna. Edible oil you will get from soya bean. So here The answer would be third option. C. The answer would be third option. Now coming to the 30th question. Cotton plant can be made to attain resistance to insects and pests by morphological adaptations, physiological adaptations, transfer of insect specific genes, all of these. So you can produce nectar-less cotton balls and smooth leaves, and also you can transfer the cry genes to produce GM crops. So all three are correct. So D would be the answer. All of these. Now the 31st question. Incorrect statement regarding Krebs cycle. Krebs cycle has 4 oxidation reactions. One turn of TCS cycle gives 3 CO2 molecules. It occurs 2 times for 1 glucose. Alpha ketoglutaric acid is a 5 carbon compound. Krebs cycle has 4 oxidation reactions. Correct. Is a correct statement. One turn of Krebs cycle doesn't give 3 CO2, it gives 2 CO2. It occurs 2 times per 1 glucose. Correct. Alpha ketoglutaric acid is a 5 carbon compound. Correct. Then the incorrect one here is B, 31B. Now the 32nd question. A cellular agent having both phosphodiester bonds and peptide bonds. Phosphodiester bonds and peptide bonds. Phosphodiester bonds are present in nucleic acids. Peptide bonds are present in the proteins, as well as if it is considered with virus or viroid, it is nothing but the caps. So both are there. That means phosphodiester bonds and peptide bonds in the sense nucleic acid and protein both will be there. That will be in virus. So 32A would be the answer. Now going further, match the following question. The 33rd one. So now we have to match this with respect to the list 2. So here disarmed TI plus. So it is a vector used to transfer desired genes into plant cells. So A will go with 4. Disarmed retroviruses. The vector used to transfer desired genes into animal cells. Microinjection method. Directly the rDNA is introduced into the nucleus of animal cell. C. Gene gun method. They are bombarded with high velocity gold or tungsten particles coated with the desired genes. This is D. Heat shock method enables bacteria to take up the recombinant DNA. E. So now the appropriate answer would be A with A with 1 minute. A is supposed to be vector U A with 4, B with 5, C with 3. A with 4 can be B with 5. So it will be third option. It will be third option. We can go to the next question. Now the question here is a bacterium divides every 35 minutes. A bacterium divides every 35 minutes. If the culture medium, if the culture medium containing 1 into 10 to the power of 5 cells per ml, 1 into 10 to the power of 5 cells per ml is grown for 175 minutes, what will be the cell concentration in ml after 175 minutes? So what is the time actually given here is 175. So 175 and the division takes place once in 35. So now if you do this, you will get 5. Then it will be 1. Taking 1 cell, it will give 2. 2 cells, it will give 4. 4 cells, it will give 8. 8 cells to 16. 16 cells to 32. 1, 2, 3, 4, 5. So the answer would be 32 into 10 to the power of 5. 32 into 10 to the power of 5. So 34th question, the third answer is correct. Now the next one, triticale, the first man-made cereal crop. Mention the type of hybridization through which it is produced. So it is a cross of wheat and rye. It is a cross of wheat and rye. They don't belong to the same genera also. So the answer would be intergeneric hybridization. Intergeneric hybridization. 35. Which of the following antibiotic was discovered by Alexander Fleming? So Alexander Fleming is well noted for the discovery of penicillin. So 36, third option. Given mineral ions essential for the physiological activities of plants, with respect to the functions, we have to match the mineral ions. So here, nitrogen metabolism, synthesis of oxygens, activation of IAA oxidase enzyme, formation of mitotic spindle. Nitrogen metabolism is with molybdenum. Synthesis of oxygens requires zinc. IAA oxidase activator is manganese. Mitotic spindle apparatus formation requires calcium. So the answer would be actually fourth option in sequence. 37, 4. The floral formula, actinomorphic, bisexual, calyx 2 plus 2, corolla 4, andrecium 2 plus 4, gynoecium 2, syncarpus, superior ovary. So this indicates or it represents which plant in the sense. It's the floral formula of Brassicaceae. We can clearly identify by the andrecium condition 2 plus 4 and also the calyx. So Brassica campus, 38, fourth option. So plant breeding question. This is just a memory type. So the plant breeding is used to obtain disease-resistant new crops. So choose the correct option for the missing terms A, B, C, D. So A is nothing but Himagiri, and it is the Brassica plant, Brassica variety Pusa swarnim, and cowpea, which is the variety Pusa comal, is resistant to bacterial blight, and the cauliflower is the crop for Pusa snowball K1, which is resistant to pearl blight black rot. So the answer is 1. Now the Next one is match the following in the construction of first recombinant DNA molecule. So now let us see the match the following. DNA ligase joins desired gene to the cleaved vector. This is correct. DNA polymerase produce multiple copies of the desired gene in host cell. This is also matching correctly. E. coli, host cell into which rDNA is introduced. C is correct. Restriction endonuclease is called as a molecular scissor. So A with 1, B with 2, C with 3, D with 4. C is the answer. So these are the answers for all the questions.